The importance of tau protein in the development of Alzheimer's disease. Alzheimer's disease is a neurodegenerative disorder affecting 5.7 million Americans. 1 out of 3 elderly Americans die from Alzheimer's or other forms of dementia, and the disease is predicted to keep increasing in prevalence in the next few decades. Despite this, there remains no cure for the disease, and research is still ongoing for an effective treatment to delay or prevent its development. There are various hypotheses on how Alzheimer's disease develops, and there's much debate within the scientific community as to which is the best explanation. The amyloid hypothesis proposes that beta amyloid protein fragments accumulate and form plaques in the brain, leading to neurodegeneration. However, new research suggests that abnormal accumulation of tau protein, and not beta amyloid, is ultimately responsible for Alzheimer's disease. What is tau? Tau is a major microtubule-associated protein found in neurons. It is an intrinsically disordered protein that assembles tubulin into microtubules and stabilizes their structure. The biological activity of tau is regulated by the degree of its phosphorylation. In a patient with Alzheimer's disease, tau is 3 to 4 times more hyperphosphorylated than tau in a normal adult brain. Hyperphosphorylation occurs when tau protein kinase in the brain becomes overactive due to a lack of feedback inhibition. Tau consists of repeating microtubule binding regions surrounded by N-terminal and C-terminal flanking regions. Hyperphosphorylation occurs on various serine, threonine, and tyrosine residues on the N-terminal and C-terminal flanking regions. Proteolytic enzymes recognize these phosphorylated residues and cleave the tau peptide at these regions. This truncated tau can no longer bind to microtubules. As a result, the microtubules become unstable and begin disintegrating. The unbound tau fragments eventually aggregate in formations called neurofibrillary tangles. However, it's still unknown whether decreased tau microtubule binding or tau aggregation is the cause of Alzheimer's pathology. How does tau aggregate? The specific chemical mechanism of aggregation is still under research. However, one explanation suggests that phosphorylation causes a conformational change in tau such that it folds onto itself, exposing charged residues on the outer surface of the protein. These charged residues can interact with aggregation inducers such as polyanions and oxidize to help form disulfide bonds which stabilize tau dimers and oligomers. Pathological tau undergoes multiple stages of self-aggregation. Aggregation begins with truncated, unbound tau. As previously explained, charged residues and disulfide bonds cause tau to form dimers and then oligomers. Tau oligomers are soluble in the neuron, and current research suggests that they're the most toxic form of the protein. These oligomers eventually develop into paired helical filaments, whose core consists of these truncated self-aggregating tau fragments. Paired helical filaments eventually aggregate to form neurofibrillary tangles that are characterized of late-stage neurodegenerative disease. Because paired helical filaments and neurofibrillary tangles are insoluble, it's unclear whether they have a positive or negative effect on the neuron. The tau fragment in the oligomeric state is resistant to proteases and is actually soluble enough to diffuse across synapses to neighboring neurons. The oligomers can then initiate the same pathological tau aggregation cascade into healthy neurons. So while hyperphosphorylated tau has been shown to catalyze and propagate the conversion of normal tau to the pathological truncated form, the process does not require abnormal hyperphosphorylation or any other post-translational modifications once it's been initiated. Experiments in solution and in vitro have shown that introduction of oligomeric tau fragments can produce pathology even in the absence of hyperphosphorylated tau. A small amount of isolated truncated tau proteins can perpetuate tau aggregation across many neurons. This could explain why Alzheimer's is a progressive disease. Researchers are looking into many possible treatments at each step of tau pathology. So far, work has been done on microtubule stabilizing agents and modulation of tau phosphorylation, but these attempts don't directly address the toxic aggregation of tau fragments. Other approaches do not depend on hyperphosphorylation theory and instead try to inhibit the self-binding of tau fragments. However, these tau aggregation inhibitors are typically based on neurofibril formation, which occurs fairly late stage, and there is a danger of the fibrils dissociating into toxic oligomer form. More promising treatment methods directly target toxic tau oligomers or try to inhibit proteolysis of normal tau into truncated fragments. A clearer understanding of the biochemical mechanisms underlying such phenomena could greatly aid in the search for a cure to Alzheimer's disease. At the very least, it looks like we're going towards the right direction.